The overall goal of this procedure is to introduce a technique which ensures cell modification with microRNA and, in parallel, cell magnetization.
This method can address key challenges in the regenerative medicine field, such as genetic engineering of cells prior to transplantation. In addition to efficient cell modification with microRNA, they are loaded with iron oxide. This can be used for enhancing cell retention in the site of interest by magnetic field application.
Cell therapy is very promising for the treatment of heart diseases. However, cell retention and cell survival urgently need to be improved.
Begin this procedure with human umbilical vein endothelial cell culture and preparation of transfection complexes as described in the text protocol. 48 hours prior to transfection, seed the HuVex on plastic cell culture well plates of suitable size with a starting cell density of approximately 13,000 cells per square centimeter of growth surface. Next, prepare fresh microRNA polyethylene imine superparamagnetic iron oxide nanoparticles. First, dilute 2.5 picomoles of microRNA per square centimeter of cell growth surface in 5% glucose solution to obtain a final concentration of 0.125 picomoles of microRNA per microliter. Next, dilute the PEI in an equal volume of 5% glucose solution. Add the diluted PEI to the microRNA solution and vortex the obtained mixture for 30 seconds. Then, incubate the microRNA PEI mixture.